Hello, my name is Megan Blanton, and for my final project, I decided to focus on the Jill Dowen Lab. The Jill Dowen Lab is a research lab here at UNC in the Department of Biology that studies how our genes are organized and how they work. One of their main projects looks at how loops in our DNA help control which genes are turned on or off. Today, I'll explain some of the techniques they use in their research. In the lab, scientists use methods such as ChIP-seq, chia-PET, and CRISPR to study how genes are controlled in cells. These tools help them understand how certain parts of our DNA can turn genes on or off. The first technique from the Jill Dowen lab that I'm going to describe is called ChIP-seq. It is used to see how proteins stick to DNA. Scientists first use a chemical to make proteins stick to DNA inside cells. This helps keep the natural interactions between proteins and DNA. Then they break the DNA into small pieces using sound waves or enzymes. They use special antibodies to pull out the pieces of DNA that have proteins stuck to them. Finally, they read the DNA pieces using a machine that can sequence DNA. This helps them find out where the proteins are sticking to the DNA. The second technique from the Jill Dowen lab that I'm going to describe is called chia-PET. This helps scientists see how different parts of our DNA are folded and looped inside cells. They use a chemical to make DNA stick together, then break it into pieces. They use antibodies to pull out pieces of DNA that are close together. They join these pieces to make new DNA fragments and read them to see how the DNA is folded. This helps them understand how different parts of the DNA interact with each other. The final technique from the Jill Dowen lab that I'm going to describe is called CRISPR. This tool acts almost like scissors that allows scientists to change DNA. First, scientists design a guide RNA that corresponds to the DNA sequence they want to change. This guide shows a protein where to cut the DNA. Then, the protein cuts the DNA at the targeted spot. The cell then tries to fix the cut by adding, removing, or changing parts of the gene. This allows scientists to make precise changes to the DNA. To summarize, the Jill Dowen lab at UNC uses ChIP-seq, chia-PET, and CRISPR to study how genes are controlled in cells. ChIP-seq allows scientists to identify where proteins bind or stick to DNA. Chia-PET allows scientists to identify interactions between proteins and DNA and better understand the structure of the gene. CRISPR allows scientists to edit pieces of DNA. Thank you so much.